Welcome to the Brain Fix podcast discussing all things brain. I'm Rob Lucas, Professor of Neuroscience here at the University of Manchester, and delighted to welcome to the podcast Lane Brown from Washington State University in the US. Welcome to the podcast, Lane.
Thank you. It's great to be here in Manchester. It's my first time in Manchester.
Well, it's a pleasure to have you. So we've heard in an earlier version of this podcast about the importance of circadian rhythms as a way of producing time in our body, and how also critical it is that those circadian rhythms are set to local time using the light-dark cycle. And you study the mechanisms in the retina that are responsible for that process, don't you?
Yes. So for about 25 years now, we've been looking at what are called intrinsically photosensitive retinal ganglion cells. And they were discovered around the turn of the 21st century. And they're the cells that are responsible for synchronizing our body clock to the solar cycle. So for maybe 50 plus 100 years of retinal research, we'd been aware of the rods and cones. And these are the classical photoreceptors that are responsible for image-forming vision, how we perceive light and motion and color and objects and things like that. Whereas the intrinsically photosensitive ganglion cells just act more as a sensor of ambient light. I like to make the analogy to a camera where the rods and cones are actually forming your image like the 20 megapixel sensor, whereas the intrinsically photosensitive retinal ganglion cells act more as a light meter. They tell your camera what the ambient light level is and how to set your exposure time and your sensitivity of your detector.
So like a key part of the eye, just like a light meter is a key part of your camera.
Yes, exactly.
And so building up on that analogy, can you give us some idea of how many rods and cones there would be in a mammalian retina compared to ipRGCs?
So in a mouse retina, there's about 13 million rods compared to about 4,000 intrinsically photosensitive retinal ganglion cells, or ipRGCs. In the human retina, there's probably 100 million rods and cones and maybe a few thousand ipRGCs. As we were discussing earlier, I think there's probably a lot of types of ipRGCs in the human retina that we just haven't detected yet because some of our methods for detecting them aren't sensitive enough to see all of them. So as we use more and more sensitive methods, I think we're going to discover more and more types of ipRGCs in the human retina, more similar to mouse.
Interesting. But I mean, at a first approximation, if you were to say in a retina what detects light, you'd say rods and cones.
Rods and cones.
Yeah. But that doesn't mean that these ipRGCs aren't important just because there's few of them.
No, they're actually phenomenally important. And they just regulate many, many processes in addition to synchronizing our circadian clock to the solar cycle. And we can talk about that more as we move forward.
Well, why don't you tell us a little bit about what they do other than our circadian clock?
So these cells were originally discovered in the late 1990s, and it was a nice combination of human research. In Chuck Seisler's lab, they discovered that some human patients who had no conscious perception of light or their environment around them visually were still able to suppress their melatonin suppression in response to a pulse of light. So that gave us the first hint that maybe there was a novel type of photoreceptor in the human eye that we weren't aware of. And then a few years later, it was actually some of your work in a mouse model where all of the rods and cones had degenerated and they were still able to synchronize their circadian rhythms to a pulse of light. And that was what really convinced me that, yes, indeed, there must be a totally unknown type of photoreceptor present in the mammalian eye. And when they were first discovered, they were thought to be a relatively simple, homogeneous population of cells that had relatively straightforward, simple functions. They synchronized our circadian clock to the solar cycle of light and dark to regulate all of our circadian rhythms like sleep and wakefulness and hormone production and things like that. And they're also responsible for controlling pupil size. So we're all aware that when we walk out of a dark movie theater or into a dark movie theater out of bright light, we can't see for the first few minutes. until our pupil dilates to let in more light. And so these ipRGCs are responsible for controlling the pupil size. As people have started studying these more over the last decade or so, many, many more functions of ipRGCs have emerged. So now they're known to be responsible for regulating mood, regulating learning and memory, regulating metabolism, even some odd things like myopia, hair growth in mice is regulated by the sympathetic system via ipRGCs, and even very odd things like contagious itch in mice. If a mouse sees another mouse scratching, it will start scratching itself, maybe like humans supposedly have contagious yawn. So I wonder if a contagious yawn is mediated by human ipRGCs. So a lot of varied but very important functions are emerging for all of these ipRGCs.
It feels like these are really influential part of our retina and play important roles in lots of aspects of our biology. What do you think links all those things that you talked about? So when you say that they're important for metabolism, for example, what do you mean by that? Why would our retina be involved in metabolism?
So I think, so some of it is linked via the suprachiasmatic nucleus, which is the home of the biological clock. And that's the brain target for the first type of ipRGCs that was discovered. But now that we have better models to look at where the more rare types project, it's finding that they have at least 20 or 30 different targets throughout the brain. And they go to centers in the brain that regulate mood, centers in the brain that regulate, either directly or indirectly, centers in the brain that regulate glucose metabolism, for instance, and indirectly to areas of the brain like the hippocampus that regulate learning and memory. So their functions fan out widely throughout the brain, both directly and indirectly.
Right. Well, that begs several questions, but I'm going to ask the one that's probably of most relevance to most people most of the time, right? What does that mean if we've got a photoreceptor which is regulating glucose metabolism? What does that mean for my life and what I should think about in terms of light, for example, and what it might be doing to me?
Well, a lot of other podcasters, one of Andy Huberman Lab's favorite sayings is that you need to see natural light first thing in the morning, and that will set your body up for the rest of the day and how it functions, because that's really what resets your clock is detection of some sort of natural light first thing in the morning to reset your body clock and start all your metabolic processes moving forward. So I think it's really important to get out and somehow see some natural lighting really early in the morning as you wake up.
Right. So that light, thinking about light as a general cue that our body uses to establish the right kind of right kind of properties for being at a daytime state, for example, is probably quite an important thing.
Right. I think you need to have your metabolism in sync with you're about to start eating. You're about to start taking in food, so you need to have your metabolism prepped to deal with that food. You need to be able to regulate your glucose properly because you haven't been eating for the last 8 hours at night. So I think having everything primed, and that's one of the functions of our circadian system, is to anticipate what our upcoming activities are and to prepare our body to be ready for that.
I guess just before we leave that, it's important for us to acknowledge that these are kind of concepts that we advocate research on at present rather than necessarily in terms of, for example, a way of regulating your glucose levels. There are much more established physiological mechanisms for doing that.
Obviously.
So
Obviously.
it's sort of a lifestyle thing you might want to play around with yourself rather than a medical intervention at this point. Is that fair?
That's more than fair. Yeah, I think there's things that we have to start considering. I think circadian considerations in medicine are going to be incredibly important as we move forward. I mean, for instance, cancer drugs are much more effective when they're taken during the right part of the circadian cycle, because either they're not broken down quickly or they're metabolized into their active form. And that happens much more efficiently if we have the right enzymes being produced and we take the drugs at the right time. So I actually think circadian biology is a very underappreciated part of medicine that we really need to spend more time looking into.
Very, very neat. Very neat. So if we say that there's the potential at least for these ipRGCs to have such wide-ranging effects on our physiology, why do you think they were only discovered 25 years ago?
Well, I think we weren't looking for them, for one thing.
Right.
It wasn't until we had these clues from Chuck Seisler's work that there were a third type of photoreceptor. But actually, interestingly enough, probably 50 plus years ago, people noted them in several papers. They just thought they were kind of a weird finding and brushed them aside because it didn't fit with the current dogma. And I think that's one of the things we have to be really careful about in science, is that if we get an odd result, don't just brush it aside. Actually pay attention to it. And yes, we need to do all the controls and make sure it wasn't just a mistake or a failed experiment. But if it passes all that vetting process, then we really need to pay attention to it, and maybe we need to alter our way of thinking and change the paradigm to take account for the new information.
No, it's really interesting because, you and I were both in the field around that time when we were trying to persuade the vision scientists and ophthalmologists that there was something in the retina that they hadn't considered. And there was quite a healthy level of skepticism about that. Is that fair?
That's very fair, but it all tumbled really quickly.
It did, yeah.
Within like 2 years, it went from a lot of skepticism to being true believers.
Yeah.
And there was just a avalanche of information that came out. Your work with the retinally degenerate mice, and Iggy Provencio cloned the gene for melanopsin, which is the photosensitive pigment in these ipRGCs. And then mice were made by Samar Hattar and King Yao's lab that showed that they projected throughout the brain. So we actually had a grant, and between the first submission, where everybody was skeptical, to the resubmission when it finally got funded, they said, well, yeah, of course we already know this. Whereas the first reviewers were skeptical. So everything tumbled and all came together really quickly in about 2 years.
So it's quite a nice story about how science can change and accept new ideas if the evidence is strong enough.
Yeah.
And in a way, it's totally understandable that people who had been studying the retina their whole life and could explain everything they observed through rods and cones said, well, I don't need to find another photoreceptor to explain the retina, so therefore I'm going to prove to me that it's right, So
right.
It did work quite well, I think, really.
Yeah, it all came together really well, and I had been studying phototransduction and rod photoreceptors starting with my PhD work, and never really considered that there was a third type of photoreceptor that we weren't looking for.
Yeah.
Until I saw this data in the late 1990s. And then I jumped on the bandwagon. And as I said earlier, David Burson was the first one to get an intrinsic light response from these ipRGCs. Well, we got our first light response 2 weeks before his science paper came out. So I guess we're number 2.
Well, that's another thing that's really important to say about science, right? It really is about reproduction, right? There's a massive... We have to acknowledge that one single observation is never enough to overturn an understanding, right? That has to be built on by our colleagues and.
Yeah, I totally agree, and even better is if findings can be confirmed by multiple independent methods.
Definitely right.
Which is the way to go for science.
Yeah. You talked a little bit about your history in the ipRGC field. Can you tell us a little bit about your personal journey as a scientist, maybe to end?
Sure. So I started off, went to the University of New Hampshire, and I've kind of known that I wanted to be a scientist ever since I was a young kid.
Oh, wow.
Um, I took organic chemistry in 10th grade or something like that. And my dad was a professor at a community college, so I thought, yeah, that's a great lifestyle. You get 3 months a year off.
Was he a scientist or?
No, he was a math professor.
Okay. Mhm.
So he taught liberal arts math, and I thought, this looks like a great lifestyle. So I went to undergraduate and got a degree in biochemistry, and then I proceeded to go to work with Lubert Stryer at Stanford University, where I first started studying the retina and phototransduction, biochemistry of phototransduction and rods, and proceeded to study cyclic nucleotide gated channels, which generate the electrical response in rods, and then kind of proceeded through the retina to study bipolar cells and then these intrinsically photosensitive retinal ganglion cells. And what I realized when I was about 45 or 50 was that you really didn't get 3 months a year off. That's when you were supposed to be doing all your research.
Yeah.
So I had overqualified myself, and it was my midlife crisis revelation was that I really wanted to be a climbing bum and live in a van down by the river. But so I've managed to balance my extracurricular activities along with a relatively productive scientific career.
Well, you're in a great part of the world if you like climbing, right? There's
Yeah, the
plenty close by.
Gritstone
Yeah.
and sea stacks, which I've never climbed in England
Yeah,
before.
yeah. No, it's a bit. There's a great history of climb. I'm terrified of heights, so I'm the wrong person to talk to. But we've had several people in the group who are climbers,
Wow.
and there's lots close
Well,
by.
this is one of the first trips I've taken in the last 20 years where I did not bring climbing gear with me.
Oh, really? What a shame.
I usually carry around a 50-pound bag of climbing gear everywhere I go, but not this time.
Do you think climbing makes you a better scientist?
I think climbing clears your mind of all your distractions, so you come back much more focused. And also, the great thing is that one thing about climbing is it's overcoming personal challenges
Right.
and figuring out how to move up the rock that you can make forward progress without falling. So it gives you a lot of confidence that if you can overcome that challenge, there's very few scientific challenges that you cannot overcome. So in some ways, it's just a real empowering experience and a confidence builder.
Yeah, maybe there's something generally there about having multiple things going on in your life and how important that is in terms of learning from your wider life for your professional activity, which is I think
I
everybody would agree with, right?
Yeah, I mean, I'm very much into having a work-life balance
Yeah.
and sometimes a work-life unbalance.
Well, well, I mean, that's been fantastic, Lane. I've really enjoyed it. I feel like we've covered a lot of ground, and it's been really fun. So thanks so much for joining us.
Yeah, this has been great. This is my first podcast, so
Right.
it's been very enjoyable. Thanks for having me.
No problem. You're a total natural. So this has been the Brain Fix podcast. Any feedback on this or future content or sponsorship inquiries, please send to the DNS inbox, which is dns@manchester.ac.uk with podcast in the subject line. And please do tune in next time for more neuroscience.